Okay, good morning, everyone. Today we are going to see things that are associated with enzymes once again, and we are going to focus on the active site of an enzyme. Now, in the last lecture, we have seen some basic definitions of enzyme. We have already seen what is the active site, but I'll just quickly revise. Active site is nothing but wherein the substrate would bind and the activity of enzyme would take place, and ultimately it gives a final product. So let us understand the active site of the enzyme. Now, if you see this, this is the binding site. This is the enzyme you can see. This is the binding site of an enzyme. Now, this binding site of enzyme is also called as the catalytic site, wherein the interaction of the substrate to an enzyme would take place. So, this active site is also called as catalytic, where the process of catalysis would take place in this site itself. Therefore, it is very important. Now, this active site of an enzyme is that point where the substrate molecule binds for the chemical reaction happens. If you see again this diagram itself, in this diagram you can easily see that this is the active site and this is the substrate that has been bind to this particular active site of an enzyme. It is generally found on the surface of the enzyme, and in some enzyme it is a pit-like structure. Pit-like structure. It means it is a dense-like structure. It is a small hollow portion that comes in the active site of an enzyme. Now, the active site takes up relatively smart of part of the total volume of the enzyme. So, total enzyme will always not work. But the active site of enzyme is said to be one of the important part wherein the interaction of molecules will take place. Active sites are cleft or crevices. It may be in this fashion or it may be in this fashion also. Substrates are bound to enzymes where multiple weak interactions, maybe very weak sort of interactions will be there between that substrate and the binding of a particular enzyme. Specificity of binding depends upon precisely defined arrangement of atoms in an active site. Absolutely, every active site will be specific for its substrate. It will not happen that any substrate would come and bind to any enzyme. No, every enzyme will be specific for binding of a particular substrate only. So configuration of that enzyme or that active site will be very specific in terms of binding of a particular substrate. The active sites of an enzyme are cleft with the enzymes which consist of few amino acid residues only. You can see these are the amino acid residues that are present. The amino acid residues itself, they fold. This is unfolding way. Now, once they fold, they get accommodated inside the active site. Now, what is the catalytic site? Responsible catalytics, it determines reaction specificity. Binding site responsible for binding of enzyme with substrate and determines the enzyme specificity. It means inside the enzyme itself, there will be some site which is called as the binding site, and another site will be called as the active site of an enzyme. So binding site will be helpful for binding of the substrate, while catalytic site will be helpful for taking the reaction of catalysis. Now speed. Properties of enzyme. Now let us understand the properties on what basis the catalytic activity or the active site works. Speed. Catalytic efficiency 10 raised to minus 3 to 10 raised to 15 turnover number of 100 days with 36 lakh per second. Specificity highly specific. Every enzyme is said to be very much highly specific. For one of the example is urease or glucokinase. Stereospecificity. Some isomers are also there which have stereospecificity. It means more than one substrate with different chemical properties to bind to a single enzyme molecule as well. Then, the example that they have taken is maltase catalyzes the hydrolysis of alpha glycosides but not beta glycosides. So, the specificity is there. Typical catalysis reactions of one stereomer of a given compound. For example, D but not L, L but not D means specificity of an enzyme would be very much in an interactive way. Reaction specificity. Some substrate can undergo different types of reactions. Each catalysis was separated in enzyme. Substrate specificity. Absolute substrate specificity. It means what is the shape of that particular enzyme and how the substrate is fitting it. It is very much specific. The example is glucokinase, urease. Relative substrate specificity. Peptane or chymotrypsin. Broad specificity, hexokinase. So these are all specificities that are associated with interaction of enzyme and the particular substrate. Now, what are the models that are associated with the active site of enzyme? The one best known model is Fisher's key model and lock and key model theory. Now, Fisher was a particular scientist and he has proposed certain model based on how the interaction of enzyme and substrate takes place. So according to this model, the active site is a rigid portion of the enzyme molecule and its shape is complementary to the substrate like key and lock. You can see the key and lock. So every key has a specific lock to be fitted in. You can, if you have a car key, it cannot be fitted into a bike key. Correct. So it is very specific towards its substrate, and therefore the enzyme and substrate reaction are specific towards each other. This is proposed by the theory. The complementary shape of a substrate and an active site favors tightly bound enzyme. So once the enzyme fits or the substrate fits into the enzyme's active site, it is very tightly built. The enzyme substrate complex formation takes place followed by catalysis. Of course, when the reaction takes place, that is ES complex development takes place, the reaction will be catalyzed and the substrate will be degraded. You can see here, this is the active site of an enzyme and the substrate is fitting up in a very specific manner, just like what a lock and key hypothesis. Another theory was put forward, that is Coastline induced fit model theory. The active site is flexible, let us say. Why it is flexible? Because in the enzyme molecule, the amino acid residue that make up active sites are not oriented properly in the absence of substrate. So this model explains that the binding of the substrate will certainly modify the active site of an enzyme. Let us see. The molecules are unstable in the induced conformation and returns to its native conformation in the absence of a substrate. When substrate combines with it, so this point is said to be one of the important part, that when the substrate combines with the enzyme, it induces conformational changes in the enzyme molecule in such a way that the amino acids that make active site are shifted into correct orientation to favor tightly bound enzyme substrate complex followed by catalysis. It means that this theory explains that the active site of enzymes, the molecules are not being arranged in a proper way. Once the binding of substrate takes place, the orientation of the substrate helps that enzyme molecule or that active site of amino acid to configure themselves in such a kind of way that they bind in a very perfect manner. The structure of enzyme. What are the structures? Scaffold to support and position active site, binding orientation substrate, catalytic site reduces chemical activation energy. So these are all specificity of a particular enzyme. The structure or defining of a structure of an enzyme is very specific. So what are the forces that are available in the active site of an enzyme? It may be electrostatic forces, hydrogen bond, van der Waals forces, non-covalent bonds. Now you can see all these 4 different types of bonds which are associated with the substrate are said to be weak bond. Why they should be weak bond? There should be weak bond because once the substrate binds to a particular enzyme and it is been degraded into product P, then that active site again the active site has to be weakened again. If the substrate has a very strong binding that is associated with the enzyme, then certainly it will not leave that enzyme or the substrate will not be degraded in a proper way. Therefore, all these bonds are said to be weak bonds. The model of, again, the induced fit and it has been shown in the diagrammatic manner. You can see here the substrate, active site of enzyme, ES complex development takes place. In the induced fit model also, you can see easily it is been. Now, if you observe this structure of an enzyme, the substrate binds here. Substrate has an orientation which will be totally different. But this substrate, once it binds, the development of the enzyme takes place in that way. The properties of affecting binding. What properties may affect binding? Complementarity, flexibility, surface, non-covalent forces, and affinity. These are things based on which the binding of a particular substrate to an enzyme, basically the active site of enzyme, will take place. This mechanism of catalytic active site, you can see the substrate. Once the substrate has been binding, this is the transition state. On the transition state, easily there will be the binding of enzyme would take place, saturation of all active site would take place on the on that basis, and ultimately degradation process would take place, and it leads to formation of a product. Okay. So that's all for today. We have learned something that is associated with the active site of a particular enzyme. Tomorrow we'll again join with another excellent topic based on enzymes.